अस्सलाम वालेकुम स्टूडेंट्स, होप यू ऑल आर फाइन। स्टूडेंट्स, आज हम करेंगे बायो फर्स्ट ईयर चैप्टर नंबर 1 टॉपिक नंबर 8 विच इज साइटोप्लाज्म एंड ऑर्गेनेलिस। द लिविंग मैटर ऑफ अ सेल इज कॉल्ड प्रोटोप्लाज्म। कि कह रहे सेल का जो लिविंग मैटर होता है उसको हम प्रोटोप्लाज्म कहते हैं। इन यूकैरियोटिक सेल्स इट कैन बी डिवाइडेड इनटू टू पार्ट्स, साइटोप्लाज्म एंड न्यूक्लियस। First of all, we discuss cytoplasm. Cytoplasm is the region between nuclear membrane and plasma membrane. बच्चों हमने प्लाज्मा मेम्ब्रेन पढ़ी है, सेल मेम्ब्रेन जो सेल की आउटर मेम्ब्रेन या कवरिंग होती है। न्यूक्लियर मेम्ब्रेन होती है जो मेम्ब्रेन होती है अराउंड न्यूक्लियस। न्यूक्लियर मेम्ब्रेन और प्लाज्मा मेम्ब्रेन के बीच में जितना भी रीजन है या एरिया है उसको हम क्या कहेंगे, साइटोप्लाज्म। दिस इज आल्सो अ कॉमन कंपोनेंट ऑफ बोथ प्रोकैरियोटिक एंड यूकैरियोटिक सेल्स. It is about 90% water and form a solution that contains all fundamental biochemicals of life. Some of these are ions, small molecules in true solution, such as salts, sugar, amino acids, fatty acids, nucleotides, vitamins, and dissolved gases. Others are large molecules, such as proteins, which form the colloidal solution. Next हमारे पास साइटोसोल. साइटोसोल is known as the matrix of cytoplasm. It surrounds the cell organelles. In eukaryote, in prokaryotes, all the metabolic reaction occurs here. Cytosol is the water-soluble component of cell cytoplasm. The cytosol is a liquid matrix around the organelles. The cytosol is a complex mixture of substances dissolved in water. Although water forms the large majority of cytosol, the term cytosol is now used to refer to the liquid phase of the cytoplasm in an intact cell. In prokaryotes, most of the chemical actions of metabolism take place in cytosol. The cytoplasm acts as a site of metabolism and storehouse of a cell. The metabolic pathways generally occur in the cytosol, which includes protein synthesis, glycolysis, etc. नेक्स्ट है हमारे पास सेल ऑर्गेनेलिस। कि सेल के अंदर जितनी भी ऑर्गेनेलिस प्रेजेंट होती हैं वो कौन सी हैं। इन यूकैरियोटिक सेल द साइटोप्लाज्म कंटेन्स हाईली ऑर्गेनाइज्ड discrete structures which are specific for various cellular functions are called cell organelles. कह रहे हैं यूकैरियोटिक सेल के अंदर, साइटोप्लाज्म के अंदर बहुत सारे डिस्क्रीट स्ट्रक्चर्स होते हैं जो स्पेसिफिक फंक्शन परफॉर्म कर रहे थे, उनको हम कहते हैं ये ऑर्गेनेलीज हैं. The organelles are generally enclosed by membrane except few such as ribosome. कह रहा है मोस्टली जो ऑर्गेनेलीज हैं वो मेम्ब्रेन से इनक्लोज हुई हुई होती हैं एक्सेप्ट कुछ के जैसे कि राइबोसोम. The organelles in the cytoplasmic matrix of a cell are कौन कौन सी organelles प्रेजेंट होती हैं एक साइटोप्लाज्म में. एंडोप्लाज्मिक रेटिकुलम, राइबोसोम्स, गाल्गी कॉम्प्लेक्स, लाइसोसोम्स, पेरोक्सिसोम्स, ग्लाइकोसिसोम्स, वैक्यूल्स, माइटोकॉन्ड्रिया एंड क्लोरोप्लास्ट. अब वन बाय वन हम इन तमाम ऑर्गेनेलिस का स्ट्रक्चर डिस्कस करेंगे और इनका फंक्शन देखेंगे क्या है. फर्स्ट ऑफ ऑल हमारे पास आ जाते हैं एंडोप्लाज्मिक रेटिकुलम. द एंडोप्लाज्मिक रेटिकुलम ER इज एन एक्सटेंसिव नेटवर्क ऑफ मेम्ब्रेन. It is a network of membrane. The word endoplasmic means within the cytoplasm. The ER consists of a network of membrane, tubules, and sacs called cisternae. From Latin word cisterna, a reservoir of liquid. The ER membrane separates the internal compartment called the endoplasmic reticulum lumen from the cytosol. There are two types of ER. एंडोप्लाज्मिक रेटिकुलम की भी दो टाइप्स होती हैं, स्मूथ एंडोप्लाज्मिक रेटिकुलम एंड रफ एंडोप्लाज्मिक रेटिकुलम. They differ in structure and function. Most of the cells contain both type of reticulum, endoplasmic reticulum. However, some cells like skeletal muscle cells have smooth endoplasmic reticulum more, where these are called sarcoplasmic reticulum. कह रहा है कि मोस्टली सेल्स के अंदर दोनों टाइप का जो एंडोप्लाज्मिक रेटिकुलम है, स्मूथ और रफ, दोनों टाइप का एंडोप्लाज्मिक रेटिकुलम पाया जाता है. लेकिन कुछ सेल जैसे कि स्केलेटल मसल सेल्स हैं, इनमें स्मूथ एंडोप्लाज्मिक रेटिकुलम ज़्यादा पाया जाता है, इसलिए इनको हम कहते हैं, इनके अंदर जो रेटिकुलम है उसको हम कहेंगे ये सार्कोप्लाज्मिक रेटिकुलम. यू कैन सी फिगर 1.15 एंडोप्लाज्मिक रेटिकुलम के इसमें सबसे पहले एक स्मूथ एंडोप्लाज्मिक रेटिकुलम है, देन रफ एंडोप्लाज्मिक रेटिकुलम है, उसके बाद एंडोप्लाज्मिक रेटिकुलम ल्यूमन है, सिस्टर्नी है, राइबोसोम्स हैं, ट्रांसपोर्ट वेसिकल्स हैं. नेक्स्ट है आ जाता है हमारे पास स्मूथ एंडोप्लाज्मिक रेटिकुलम क्या है. द स्मूथ एंडोप्लाज्मिक रेटिकुलम इज continues with rough endoplasmic reticulum since ribosomes are not attached to it. Therefore, it has smooth appearance. अब इसको हम स्मूथ क्यों कह रहे हैं कि कह रहे हैं कि जो स्मूथ एंडोप्लाज्मिक रेटिकुलम है इसके ऊपर राइबोसोम्स अटैच नहीं होते, प्रेजेंट नहीं होते, तो इसकी जो सरफेस होती है वो स्मूथ होती है. द स्मूथ एंडोप्लाज्मिक रेटिकुलम फंक्शंस इन diverse metabolic processes. Enzymes of smooth endoplasmic reticulum are important in the synthesis of lipids, including oil, cholesterol, phospholipids, steroids, for example, sex hormones of vertebrates and various steroid hormones of adrenal glands. In the smooth endoplasmic reticulum, other enzymes help to detoxify drugs and poisons, especially in the liver cells. Next आ जाता है हमारे पास rough endoplasmic reticulum. The rough endoplasmic reticulum has ribosome attached to the side-facing cytoplasm and has rough appearance under electron microscope. जो रफ एंडोप्लाज्मिक रेटिकुलम है इसके साथ राइबोसोम्स अटैच होते हैं इसलिए इसकी रफ अपीयरेंस होती है. That is why it is called rough endoplasmic reticulum. Many types of specialized cells secrete proteins produced by ribosomes attached to rough endoplasmic reticulum. Secretory proteins depart from rough endoplasmic reticulum wrapped in membrane of vesicles that bud like bubbles from specialized region called transitional endoplasmic reticulum. Vesicle in transit form one part of the cell to another are called transport vesicles. In addition to making secretory proteins, rough endoplasmic reticulum is a membrane factory for cell. It grows in place by adding membrane proteins and phospholipids to its own membrane. The endoplasmic reticulum membrane expands and is transferred in the form of transport vesicle to other components of endomembrane system. Next आ जाते हैं हमारे पास राइबोसोम्स. Ribosomes are particles made up of ribosomal RNA and प्रोटीन. कह रहा है कि राइबोसोम पार्टिकल्स हैं जो किससे मिलकर बने होते हैं, राइबोसोमल आरएनए और प्रोटीन से. हमने बच्चों पढ़ चुके हैं कि आरएनए की तीन टाइप्स होती हैं, मैसेंजर आरएनए, राइबोसोमल आरएनए, ट्रांसफर आरएनए. दीज ऑर्गेनेलीज कैरी आउट प्रोटीन सेंसिस. ये क्या करती हैं, राइबोसोम्स का काम होता है प्रोटीन सेंसिस. Ribosomes were first observed using electron microscope as dense granules. Ribosomes are roughly spherical, granular, non-membranous, found in both eukaryotic as well as prokaryotic cell. However, eukaryotic ribosomes are large होते हैं and characterized as ATS ribosomes. उनको हम ATS ribosome कहते हैं. While the prokaryotic are slightly smaller and are characterized as SEMTS ribosome. They can be seen only under microscope. Under electron microscope, they are made up of almost equal amount of RNA and protein, so they are also called ribonucleoprotein. Ribosomes are formed in nucleolus, though then these are transported to the cytoplasm through the nuclear pore. The eukaryotic ribosomes are composed of two subunits of different size. The large one is 60S and smaller one is 40S. The two subunits on attachment form ATS particle. The attachment is controlled by presence of magnesium ions concentration or forming salt bonds between phosphate group of RNA and amino group of amino acid or both by magnesium ions and salt bonds. Both ribosomal subunits are generally attached together at the time of their function. The ribosomes are involved in the event of protein synthesis. Sometimes during protein synthesis, several ribosomes are attached to one mRNA messenger RNA molecule. Such a chain of ribosome is called polysome or polyribosome. कह रहा है कि कुछ राइबोसोम्स क्या आते हैं कि ड्यूरिंग प्रोटीन सिंथेसिस वो अटैच हो जाते हैं मैसेंजर आरएनए के ऊपर. इस जो चेन ऑफ राइबोसोम है इसको हम क्या कहेंगे, पोलीसोम और पोली राइबोसोम. In this way, several copies of small polypeptides can be produced in very less time. Next आ जाती है ऑर्गेनेली हमारे पास गोल्जी complex. It is found in all eukaryotic cell. It was first discovered by Italian biologist Camillo Golgi in 1898. It is also called Golgi apparatus and Golgi bodies. Golgi complex consists of stack of flattened membrane-bound sacs called cisternae, together with system of associated vesicles called Golgi vesicles. कह रहे हैं जो Golgi complex है ये एक stack है किस चीज़ का, flat membrane bound sacs का, जिनको हम cisternae कहते हैं. It is a complex system of interconnected tubules formed around the center stack. At one end of the stack, new cisternae are constantly being formed by the fusion of vesicles from smooth endoplasmic reticulum. This outer or forming phase or cis phase is convex, while the inner end is concave and is called maturing phase or trans phase, where the cisternae break up into vesicle again. The most important function of Golgi complex is the processing of cell secretion कि जितनी भी cell secretion करता है उसकी processing करता है. Therefore, these organelles are abundant in secretory cells के secretory cells में बहुत ज़्यादा पाई जाती हैं. Golgi complex collects these proteins from rough endoplasmic reticulum through SCR. Smooth endoplasmic reticulum modify them to perform specific function and then export these modified products in the form of vesicle. Certain organelles such as lysosome, peroxisome and glyoxysome also originate from Golgi complex. Golgi complex is also involved in the formation of conjugated molecules like glycoprotein, lipoprotein, etc. In plant cell, during cell division, Golgi complex also gives rise to vesicles which contain cell wall synthesizing materials. At cytokinesis, these Golgi vesicles are arranged on the cell equator, fuse together and form a structure called phragmoplast. Later on, new cell wall is derived from this structure. Next आ जाता है हमारे पास vesicles. The term vesicle derived from the Latin word vesicula meaning smaller bladder. Vesicles are small cell organelles which are present in cell. These organelles are small membrane enclosed sacs. A vesicle is a small structure within a cell consisting of fluid enclosed by lipid barrier. A vesicle consists of hydrophilic end and hydrophobic tail. Many vesicles are made in the Golgi complex and the endoplasmic reticulum or are made from parts of the cell membrane by other membranous material. Examples of vesicle include secretory vesicle, transport vesicle, synaptic vesicle, lysosome, etc. Vesicles are found in bacteria, archaea, and plants as well as in animals. Function of vesicle is that they transport substances to and from one cell to another and also from one part of cell to another. For example, a protein and other molecules. The first step in vesicular transport is the formation of a vesicle by budding from the membrane. Vesicles are also involved in metabolism, buoyancy control, and enzyme storage. They can also act as chemical reaction chambers. Next organelle आ जाती है हमारे पास lysosomes. Lysosome means splitting and soma means body. These are single membranous spherical vesicles होते हैं. They contain digestive और hydrolytic enzymes. इनके पास digestive और hydrolytic enzymes present होते हैं. The lysosomal enzymes are made by rough endoplasmic reticulum and transported to Golgi complex through SCR. After modification, these enzymes are released from transphase Golgi complex in the form of vesicle. Such vesicles are called primary lysosomes. The newly formed lysosomes before the start of function are called primary lysosomes. In plants and fungi, certain vacuoles carry out enzymatic hydrolysis, a function shared by lysosomes in animal cells. Lysosomes contain about 40 different digestive enzymes. These enzymes can break down every major macromolecule of the cell. The contents of the lysosomes are acidic. In order to perform its function, the lysosome fused with membrane-bound vesicles that arise from any of these pathways, endocytosis, phagocytosis, or autophagocytosis. The key function of lysosome is digestion and removal of waste. Foreign particles are pulled into the cell through process of endocytosis. The process endocytosis happens when the cell membrane falls on itself invagination, creating a vacuole or a pouch around the external contents and bringing those contents into the cell. The process by which unwanted structure within the cell are engulfed and digested within the lysosome is called autophagy. This is self-eating process of cell, in which cell lysosome begins to digest cell on organelles. कह रहे कि ये सेल का सेल्फ ईटिंग प्रोसेस होता है जिसमें लाइसोसोम digest करता है अपनी organelles को. Such lysosomes are called autophagosomes. This process either takes place in starvation period in order to obtain energy or it occurs in routine in order to control number of specific organelles. For example, if someone starts to perform heavy muscular exercise, the number of mitochondria begins to increase in the muscle cell. But if he leaves exercise, The number of mitochondria are again decreased by process of autophagy. Sometimes, especially during developmental phase, when a particular cell is required to disintegrate it, a type of cell death is committed called autolysis. This is a programmed cell death in which lysosomes burst and their enzymes contents are quickly dispersed through the cytoplasm. In this way, the cell is disintegrated into fragments which are phagocytosed by other cells. Due to this function, lysosomes are also called suicidal bags. नेक्स्ट हमारे पास ऑर्गेनेली इज peroxisome and glyoxysome. Peroxisome. The peroxisome is a small vesicle, single membrane bound organelle. It is an oxidative organelle that is not part of endomembranous system. Like mitochondria and chloroplast, it imports its protein primarily from cytosol. Peroxisomes can vary in shape and size depending on the need of cell they serve. They will sometimes increase in number and size if, for example, they have a lot of alcohol to break down. Peroxisome and glycosomes are collectively called microbodies. किनको हम together microbodies कहते हैं? They are similar to lysosomes in sense they are single membranous vesicular structures. They contain enzymes although different than lysosomes and originate from Golgi complex, but they are smaller than lysosomes. Peroxisomes contain some oxidative enzymes like peroxidases, catalases, and glycolic acid oxidases. They are abundant in liver cells. They are specifically involved in formation and decomposition of hydrogen peroxide, so they are named peroxisomes. They are mainly concerned with detoxification of alcohol. In this activity, alcohol is oxidized into hydrogen peroxide with the help of peroxidase enzyme. Hydrogen peroxide is itself a toxic molecule which is immediately broken down into water and oxygen by another enzyme called catalase. In plant cell, peroxisomes are involved in photorespiration. A step of photorespiration takes place in peroxisome in which glycolate is converted to glycine with the help of enzyme called glycolic acid oxidase. Next are the glyoxysomes. Specialized peroxisomes are also called glyoxysomes. Glyoxysomes are found only at seeding stage in oilseed plants. These organelles have a number of enzymes specific for plant lipid metabolism that are not found in animal cells. The germinating seedlings convert storage fatty acids to carbohydrate. This is achieved through a metabolic pathway called glyxylate cycle, the enzymes of which are located in glyoxysomes. नेक्स्ट आ जाते हैं हमारे पास वैक्यूल्स. Vacuoles are large vesicles that originate from endoplasmic reticulum and Golgi complex and plasma membrane. Vacuoles perform a variety of functions. Many freshwater proteins have contractile vacuoles that pump excess water out of cell, therefore maintaining a suitable concentration of ions and molecules inside cell. In young plant cells, many small vacuoles are present which may hold reserves of important organic compounds. These vacuoles may also help in protection of plant against herbivores by storing compounds these that are poisonous or unpleasant to animals. Mature plant cells generally contain a large central vacuole which develops by joining other small vacuoles. The solution inside the central vacuole called cell step is plant cell's main reservoir of inorganic salts, ions, including potassium chloride. The central vacuole plays a major role in mechanical sport by maintaining turgor and also acts as the storehouse of cells. The membrane separating the vacuole from cytoplasm is called tonoplast. Next आ जाता है हमारे पास mitochondria. Mitochondria singular mitochondrion are present in all eukaryotic cells. Some cells have a single large mitochondria, but more often a cell has 100 or even 1000 of mitochondria. The number correlates with the cell's level of metabolic activity. For example, cells that move or contract have proportionally more mitochondria per volume than less active cells. Mitochondria are capable to divide themselves in order to increase their number. They divide by fission. Mitochondria are cylindrical rod-shaped structure. They are enclosed by double membrane, the outer membrane and inner membrane. The outer membrane is smooth and somewhat like a sieve due to presence of porins. These are spherical proteins responsible for transport of molecules across membrane. Porins allow free passage of various molecules into the intermembrane space. The inner membrane is selectively permeable and folded inwards. The folds are called cristae, which serve to increase the surface area. The inner surface of crystal has granular structures called F0 or F1 particles. The particles are actually ATP synthase enzymes. In addition, carriers in electron transport chain. The inner membrane divides the mitochondria into two intercompartments. The first is the intermembrane space, the narrow region between the inner and outer membrane. The second compartment, the mitochondrial matrix, is enclosed by inner membrane. Mitochondrial matrix is a jelly-like material that contains a smaller circular DNA, all kind of RNA, ribosomes, MTS, and enzymes. It means the proteins which are required by mitochondria are synthesized by on metabolic activity. Mitochondria are the sites of cellular respiration, the metabolic process that use oxygen to generate ATP by exciting energy from sugars, fats, and other organic bonds. Enzymes in the matrix catalyze some of the steps of cellular respiration in Krebs cycle. Other proteins that function in ATP generation through electron transport chain are found into the inner membrane. Next time is plastics. On the basis of presence of or absence of type of pigment and the stage of development, plastics have been classified into three types: proplastid, leucoplastid, and chromoplastid and chloroplastid. Four types. Proplastids are young, immature, and developed plastids. They are self-replicating organelles. There are three types of plastids. Leucoplasts are colorless, found in parenchyma, cells of roots, stem, and seeds. Chloroplasts synthesize different colored pigments other than green. Chloroplasts are found in green parts of plants and act as site of photosynthesis. Structure and function of chloroplast. Chloroplast is a disguised structure which consists of three parts: envelope, stroma, and thalakite. Each chloroplast is bounded by a smooth outer membrane envelope. The outer membrane, like mitochondria, surrounds contains the porins and therefore is freely movable to small molecules. The inner membrane is permeable, rich in protein. Between the outer and inner membrane, there is semi-permeable membrane. The ground mass of chloroplast is called stroma. It is colorless, proteinaceous substance which is like mitochondrial matrix also contains small circular DNA, all kind of RNA, ribosomes, MTS, and all various enzymes. The stroma contains a stem of chlorophyll, being double membrane, flattened sac-like structures called thalakkites. There are two types of thalakites, smaller thalakite and larger thalakite. Smaller thalakites are disc-like sacs which are filled over one another like stack of coin. Each stack of smaller thalakite is called granum plurigrana. Each granum consists of 25 to 50 thalakites and there are about 40 to 60 grana found in each chloroplast. Phenotypic pigments are also found in the membranes of smaller thalakites. Larger thalakites connect the grana with each other and are called intergrana. The membranes are colorless as they do not have pigments. Chloroplast is the site of photosynthesis in plant cells. The first phase of photosynthesis is light-dependent action in which sunlight is captured and transformed into ATP. This phase takes place in granular region of chloroplast. The second phase of photosynthesis is light independent action, dark reaction, in which CO2 is reduced to make carbohydrates. The enzymes for this activity are found in stroma region of chloroplast. In figure 1.6, you can see the structure of chloroplast. Next organelles are centrioles. Centrioles are non-membranous cell organelles found only in animal cells. They are also found in fungi like proteus, such as slime mold and water mold. Centrioles are also structures and usually occur in pairs. These occur at right angle to each other nearly one pole of nucleus. Each centriole is composed of nine triplets of microtubules which are circularly arranged around a central axis. Just before the cell division, the pair of centrioles duplicate and become two pairs which later on migrate to opposite pole. Both centrioles pair give rise to microtubules, spindle fibers during cell division. The whole structure of fibers is known as mitotic apparatus, which helps in the distribution of chromosomes between the daughter cells during cell division. In addition, centrioles also give rise to basal bodies of cilia and flagella. Next आ जाता है हमारे पास न्यूक्लियस. Nucleus is the most prominent and most important part of cell. In animal cells, it is found in center, with the exception of muscle fiber cells. But in adult plant cell, it is slightly away from center due to presence of large central vacuole. A typical eukaryotic nucleus consists of nuclear envelope, nucleoplasm, nucleoli, and chromatin. Nuclear envelope, also called nuclear membrane, double membrane covering which makes the boundary of nucleus. Both membranes of nuclear membrane are separated by a fluid-filled permeable space. Thus, membranes are composed of lipid bilayer and protein. The outer membrane of nuclear membrane is covered with ribosomes and is connected with the membrane of endoplasmic reticulum. There are numerous pores in the nuclear envelope called nucleopores, which are composed of specialized transport protein called nucleoprotein. At the point of nucleopore, both the membrane are interconnected. These pores regulate the nucleocytoplasmic exchange of material. This exchange includes RNA and ribosomal proteins moving from nucleus to cytoplasm and proteins such as DNA polymerase, carbohydrates starting and moving lipids into nucleus. Although small molecules simply diffuse through the pores, larger molecules may be recognized by specific signal molecules, then be diffused with the help of nucleoporine into and out of nucleus. Next are the nucleoplasm. It is the transparent semi-fluid ground substance formed of a mixture of proteins, enzymes, free nucleotide, and some metals, ions for the synthesis of DNA and RNA. It also controls histone and non-histone DNA proteins, so the nucleoplasm is slightly different from cytoplasm. Next are the nucleolus. Nucleolus is a non-membranous bone structure. The nucleoplasm, a cell may have one or more nucleoli. Nucleolus appears during interphase and disappears during cell division. A nucleolus consists of a peripheral granular area contains ribosomal RNA and ribosomal DNA. Therefore, nucleolus is involved in the construction of ribosomes. Chromatin and chromosome. Chromatin is a network of thin thread-like structures made up of DNA and proteins. During cell division, chromatin fibers begin to condense and coil up into separate structures called chromosomes, which are thick enough to be seen in a light microscope. A typical chromosome consists of two strands called chromatids, which are attached with each other at a point known as centromere. Centromere is construction functionally related to movement chromosome during cell division. Each centromere has a complex kinetochore protein present on the opposite side of construction. नेक्स्ट टॉपिक आ जाता है हमारे पास प्रोकैरियोटिक एंड यूकैरियोटिक सेल. व्हाट इज द डिफरेंस बिटवीन प्रोकैरियोटिक सेल एंड यूकैरियोटिक सेल? Two kinds of structural differences have been evolved over time. Prokaryotic cells include archaea, bacteria, and cyanobacteria, whereas all of the other forms of life, like plants, animals, are composed of eukaryotic cells. In figure 1.3, you can see you can see difference between prokaryotic and eukaryotic cell. Table 1.2, difference between prokaryotic and eukaryotic cell. Prokaryotic cell, nucleus and other membrane bound organelles are absent. Main difference ये है कि prokaryotic होता है, pro means before, carry means nucleus के before nucleus. इनके अंदर nucleus और membrane bound organelles क्या होती है, absent होती हैं. Nucleus and other membrane bound organelles present eukaryotes में. U means true, carry means nucleus, यानी true nucleus present. Prokaryotes are always unicellular. Eukaryotes are unicellular, also multicellular. DNA is stored in cytoplasm. DNA is stored in nucleus. Prokaryotic cells have one primary circular chromosome and various plasmids. In eukaryotic cell, DNA is stored in double-stranded chromosome. Ribosome in prokaryotic cell are 70s with 30 and 50s subunits. Ribosome in prokaryotic cells are 80s with 40 and 60s subunits. So ये मेरा टॉपिक कंप्लीट हो चुका है. होपफुली आपको समझ आ गया होगा. If you have any doubt, you can ask in comment section. Please like, share and subscribe my YouTube channel. Thank you.